Jeff, do you want to share or?
Yeah, sure. Um, Before I get started, just a little big context here is I was working on some ideas and I was writing them up for the last several days. And then yesterday I read a paper, which really threw me for a loop, introduced something completely new I hadn't known about. And so I was like starting over again. So I was like the last, few hours I've been like trying to like, okay, how am I going to present this? So just it'll be even more scattered than my usual. So in some sense I'll be presenting some new results some from other people, but I'm going to try to put it in the context of what I was working on. So just a heads up that my Excalibur pictures I'm going to show you are kind of scattered because of this something I just discovered yesterday. So let's just get into it. I'm going to share my screen and I'll show you my what's that? I don't want that. I don't know who that is. Let's zoom. Here we go. Hopefully. You see my Excalibur drawing here? You
Yes.
see it?
Yep.
All right, let's just zoom over here. Uh, so let's just start with this box I've highlighted here. And, I've always said that the key to solving problems is to really define the problem clearly. And then once you define the problem clearly, the answer sort of presents itself. I'm trying to do that here. I haven't achieved that yet, but so I just thought there's a whole bunch of things we've been talking about over the last few months and I think they're related. And so I would label this as three problems that suggest we need to change our thinking about reference frames. So I just list them here. Hopefully, we'll have some memory of these things. So the first number one is shared learning. This is the idea that columns we can't expect every column to be exposed to every learning point, and we need all the columns in a region um, to have learned models. And so we have columns not getting it, but also need to learn. So we have to have some sort of shared learning mechanism. We started this idea with columns within a region. Trying to remember how do I get my little highlighter in. Here we go, like that. There we go. Columns in a region. Like, okay, so there's a bunch of columns in V1. They all have to have similar models, even if they don't expose to it. And then that got extended because we were talking about the hippocampal complex and how it and the cortex are learning models of the same object, but the hippocampus is learning quickly and the cortex is learning slowly. So somehow when I think I'm learning something in V1, I have to be learning it in the hippocampal complex as well. So it's beyond the single region. Uh, it's not clear whether this shared learning occurs across modalities. Like we think it might be like if I'm learning something via vision, am I also training the tactile column, touch sensors, and whether it applies to behavior models. Anyway, it does imply that if I'm going to be sharing some learning globally, that basically learning one feature at a time or one child object at a time, and we're sharing that with a lot of other columns in the neocortex. And this requires that multiple columns are anchoring to the same reference frame for at least a reference frame for the same object, and they're somehow sharing movement and feature data. So we didn't have that in Thousand Brains or in Monty right now. Well, not Thousand Brains. And this idea led to the idea that when we're voting, that the voting ID also anchors the reference frame somehow, so that maybe even though there's only a few columns in a region that are observing something, um, all the columns will anchor to the correct reference frame and they'll all be learning something similar. So that was one problem, the shared learning problem, which is pretty challenging. Another problem which is we've had for a long time, is voting using relative pose. And we have a solution for this in Monty, but I don't think we have a good biological solution for it. And we consider two types of solutions. One is a column a column needs to know like not just what it's observing, but somehow where it is on the overall object or where it is relative to other columns and during the voting process. And so it could be one solution could be relative to nearby columns. Like it say, hey, you're a little bit over there and I'm a little over here. Let's combine. Or it could be, we've talked about how relative to some shared attention area. Like you might have, like, oh, we all agree that this object we're viewing is in some physical space, and where am I relative to that physical space? Um, the mechanism for this seems to be dynamic. It goes on the fly, but it can put partially learned. It can be partially structural, meaning like it could be like, oh, maybe the columns on the retina have some, you know, constant displacement from each other, something like that. I don't think that's true, but we've discussed these ideas. That's another second problem. The third thing is that I don't think I've ever called it out like this before. I'll just labeling it distributed models. Now, we've always had the idea of distributed models in the Thousand Brains theory. We use distributed models for voting, right? We have multiple models of an object, and they are different, observing different parts of the object, and so we have it's not a single location. It's at multiple models. It's distributed, and we rely on those distributed models for voting. But it came clear to me that we need to extend our idea of distributed models um, to include something like prediction, which we don't have today, uh, and maybe behaviors and maybe motor output. And this became evident when I was thinking about um, how models could exist in both the hippocampal complex and in the cortex. That is, remember, with the ideas you might have a model of the intersection or an object that's put a permanent in the cortex. And at the moment when you're learning something new about it, you're actually forming most of that learning is happening in the hippocampal complex. And yet these seem to be combined. So when I make a prediction about I'm looking at the intersection that I often talk about, and now I've learned that there's a car here and a car there. Well, those particular details are going to be in the hippocampal complex. Um, but when I make predictions about the intersection, I'm combining the predictions from the hippocampal complex and the cortex. There's some seamlessly combined. So I don't even know, like, oh, that was permanent. This is temporary. Um, it just feels like, we didn't, it felt like we didn't have really a way of doing that. May not even be just describing the problem well, but I felt like, hey, that's an idea that I hadn't really thought about before. Like, how do I combine these models so I feel like I just have one? Um, and I'm not aware that it's distributed, and the distributors are like predictions. Like I'm predicting what I'm going to see at some place. So some prediction could come from the cortex, it could come from the hippocampal complex, and then it seems like, well, we might extend the same thing for behaviors and motor output. Like, how do I? Seems like there's a more general problem of doing distributed models than we've sort of internally conceptualized. At least I haven't. So I wish those are more concrete, but does anyone have any questions about these different problems? We've talked about them all in the past.
Just a note or a question about the voting ID anchors the reference frame. Are we thinking about this only in learning, or we also think that this might have a role in during inference? Once
Well,
they.
I would think it happened all the time. Um, well, I don't know. It's an interesting question. During learning, it wasn't. Let me step back. Clearly, during learning, because this idea that when you're learning, you're doing one thing at a time. That's sort of contrary to voting, right? In voting, I might have multiple columns all doing their own hypotheses, and so they all have their own thing going on. Um, but during learning, we're saying, no, we're all going to lock into the same thing. We're all going to learn this is the feature now, and this is the next feature. Um, so it's definitely during learning. I'd have to think about it more during inference. I was assuming it did happen during inference, but I have to think through it more carefully.
It's just because I'm thinking about anchoring or re-anchoring. Or usually, my understanding is that it doesn't just anchor to an object or reference frame. It has to anchor to a location in that reference frame. So it may not
Right,
be just enough to have the voting ID, but it
right.
needs
It
some
isn't
other
enough.
signal from.
Right,
Yeah.
right. You're absolutely right about that. So, um, in this little paragraph here, I say requires multiple columns anchoring to reference frame for some object and sharing movement and feature data.
Hmm.
So, so you need to somehow share the feature data as well. Like if we're all learning, and I say, oh, I've detected this, 45-degree edge here on this location. So every column in theory has to be on the same, it has to be anchored to the same object reference frame, at least their version of it. And they'd have to know, oh, what is the feature and what location am I in? I use the word here sharing movement as opposed to sharing location because the theory is that you might be able to, if everybody, um, once you do have a single location correctly, I'm a column and I'm not observing anything. But if I did know the correct location, then any movement vector that comes in, I can apply to my reference frame. So someone doesn't have to necessarily be passing me the location. I could just be sharing movement, and
Mm.
everybody's updating their locations independently. But then you have to summon, how did you know the location to begin with? So these are the mysteries, Rami. I mean, this is really confusing to me. So it's a good question.
Yeah. Yeah. Well, I can see like if it's getting the voter's ID and at the same time it's getting the feature that it's looking at and maybe some like idea of the neighboring locations at the same time, like through some cortical connections, then it may be able to like all of these signals can force it to re-anchor to some location in its own reference
Right,
frame.
right,
But
right.
I don't know how they all work together. And
Right, right. Well, that's going to get at the heart of what I'm talking about in a minute here. Um, because I'm going to jumping ahead here a bit, but the way that I've been thinking about how this works in biology is we didn't really have a way of it actually didn't have a biological mechanism to make any of this work. And I'll get into that in a second. Um, even re-anchoring, the way I was thinking about how locations are are specified in a column, it's not even clear how I would even do path integration. And I'll get to that in a
mm,
moment. Um, I mean, the nice thing is we have solutions for this in Monty, and or we have a lot of different options because Monty doesn't try to do this in a biological way. Monty's using different types of reference frames and Cartesian coordinates. So anyway, I guess it's great questions, Rami, and it's sort of getting at the heart of like why is this so hard to think about? I find it confusing. Does anyone else want to ask questions about any of these?
Maybe just to throw it out as another, like one alternative to the shared learning. I know it's not our kind of front runner, but I think it's worth keeping in mind is that like instead of thinking about all the columns need to be learning about the object at the same time, one option could be that a column high up in the hierarchy who can learn multimodal representations quickly learns the object representation and replays that later to lower regions to kind of like the kind of model transfer lower down in the hierarchy instead of learning at every level at the hierarchy at the same time, just at different speeds.
Yeah. Well, I agree with that. That could definitely happen. But there's a little mystery in that whole too, right? It's like, oh, some region higher up that's doing multimodal. I don't know how to do that either, right?
Yeah,
That's sort of like,
it would still require anchoring the reference frames and sharing movement and feature data.
right.
So
It's
it
sort
would
of
just
like,
not have to happen during the
right, right.
actual observation of
So
the
I
object.
have the same basic problem, but maybe we could divide and conquer. Somebody has to solve this problem, then later we could, you know, it's
Yeah,
like it's an added possibility on top of the basic problem, I
yeah.
think.
And
Yeah.
maybe the stuff you're going to discuss will kind of get at this as well. But maybe like another problem that feels like it could fit in here is just the sort of the question of like how do we fit sufficient grid cells or grid cell modules into cortical columns? Because the entorhinal cortex is pretty big. In general, if we want like unique location codes, we normally want a lot of grid cell modules. And
Right.
so that's like, okay, biologically, how does that fit into a column? And
Right. Right.
so
Right.
so
Right.
that's like
Well,
another
right.
kind of thing. Like, does that mean are we thinking about it kind of slightly the wrong way?
Well, I
Um,
think it's funny because, all right, I'm going to talk all about that in a second. So,
okay.
uh, and maybe you and I have a slightly different understanding where we were. Um, Maybe I should do that right now. Let's start with a little history. Can we do that? Um, a little history here. Uh, a long time ago, we deduced the need for locations and reference frames in cortical columns. That was a hard deduction. Yes, it must be happening because columns can make these predictions and they need to know where they are. And the different columns make different predictions. But we then make the assumption that there was a mechanism similar to grid cells that existed in all columns. So that was just an assumption, a guess, and it just seemed pretty solid guess because wow, good cells are really complicated, and it'd be unlikely the brain would come up with something else. And the hippocampal complex is just an extension of the cortical sheet. So all these reasons suggested that, and now we know there's evidence for this. So this is pretty clear that something like that's happening. Then the problem presented itself is that a single grid cell module, and this is what you were just talking about, has very limited resolution. It's basically, it can represent like 20 or 30 or 50 different points in space, and then they repeat over and over again. Therefore, we were going to go with this idea that we didn't come up with, which is to rely on multiple grid cell modules to represent space. Now, I don't know. I don't have a picture of this, but well, actually, I do. This is from one of our papers, and here you can see this is one of the figures, and the idea is look at the C, the two grid cell modules in C. These are two grid cell modules. Each has a sort of limited resolution. That is, when you see the red cell active, it could be any one of these points that you're in the space. And if you see the green cell active, it could be any one of these points. So that's not very specific. But if you combine the red and the green, and then at any point in time, you'll have some coincident activity, and that's a much more accurate position of where you are. So this is an idea of combining, and that's, I think, what you were just referring to, Niels. And so we, in this paper from us back in, I forget what year it was, we went with this. Other people came up with this idea and said, well, this is brilliant. This is really, I love this idea. And if you had enough grid cell modules, and each one just, all it has to do is has anchor differently and have a slight different orientation, which is what we see in the real world, then you can have a very high resolution space. And each grid cell module does path integration on its own. And so we have the best of all worlds. So we went with this idea in that paper. The problem was, so there we go. This worked well. And he said, well, if we have 20 more grid cell modules, that we create a very large space. You need some number of them to make this work well. And then path integration works from any location, which is wonderful. And then by remapping each module, defines a region of space dedicated to a single object. So it's like the grid cells, had a bunch of grid cell modules. They had this huge, size of the universe space. And then by remapping each module, you define some region of that space, and that would be the model that you'd use that would be a particular object's model space. And path integration worked automatically, which is great. But then there was a problem. In the entorhinal cortex, there are less than 10 grid cell modules, and they are separate and they don't interact. So I'll get to that in a second. And in the cortex, it was hard to see how multiple grid cell modules could coexist in a square millimeter column. So if we go over here, this is like how the entorhinal cortex looks. You might have something like, it's less than 10. You might have, and so this is some recent data I'm going on here. A grid cell module is maybe a millimeter squared, and they're arranged in an order. There's less than 10 of them. So here I show 7. They go from on the dorsal side, one end of the entorhinal cortex, to small resolution, and the other one having very large resolution on the ventral side. And I talked to a bunch of neuroscientists about this, and I asked them, is there any evidence that these things are integrated together? Like, does any cell up here in the hippocampal complex get inputs from all of these? And the answer was no, they don't. And I asked, do they overlap? And the answer was no, they don't. Oops, oh shoot, sorry, this happens all the time to me. I just skipped to another page. Here's what I want here. I have to be careful when I scroll around. It often does this. Let me just zoom out here a bit. I want to show you this picture. You may remember this picture. This is from Tank et al. And this was showing a grid cell module in the entorhinal cortex. And he was the first person I know who sort of mapped it out really nicely. So this is one grid cell module here. And then this would be at one scale. And then this blue one was the next grid cell module. and going from, ventral to dorsal. And this was at a different scale. And I asked him, is there any overlap? He said no, and so on. So everyone showed these as sort of separate entities. We can go into details, remind you what's going on inside of this, but basically within a grid cell module, you actually have, he was showing multiple, what he calls phaselets. That is, you would have multiple bumps of activity inside a single module, not just one. So there'd be multiple cells that represent the same point. It's just like you take a grid cell, what we think of as a grid cell module, and make 6 of them. So at least you have some redundancy here. And so there'd be multiple bumps of activity moving around on this. Anyway, let's go back out again. Um, uh, to here. I got it. All right. So the problem we had was there are less than 10 grid cell modules. That wasn't enough. And they're separate and they don't interact. And in the Cortex, it was hard to see how I could get multiple grid cell modules could exist in a column. So maybe the solution the way I've been thinking about this for a long time now, and Niels, your comment earlier made me think that maybe we didn't understand this together.
No,
That I assume,
yeah, I think it's consistent with what you've written here.
okay,
Anyways, yeah.
I assumed I had a sort of temporary assumption here, the solution. I assumed that there was a single grid cell module per column. That's what it looked like in the entorhinal cortex. And that we were going to use mini columns to create the unique SDR for each grid cell. So this is similar to what we do in layer 4 and layer 3B. That is, you take a simple representation, you have a mini column of cells, you pick a single cell in each mini column, and now you have a very high unique SDR. And so that's nice. That would form a very unique location, even if I only had a single grid cell module, because each grid cell would have, I'd pick one of n cells, and you'd have a very unique SDR. There were problems with this. Path integration and remapping were problematic. Like I have to somehow do this at the cellular level, and I had no idea how to do that. And so we didn't resolve these issues. I've talked about them in the past with possible solutions and so on. Monty doesn't have this problem because Monty uses Cartesian coordinate reference frames. So just a picture of what that looks like here. So the solution I came up with is shown here. I said, well, let's assume there's a one grid cell module. This is represented by this upper rectangle here, and this is the bump of activity on the grid cell module. So I'm not showing it accurately. And you assume you have just one of these modules per cortical column, and then you have a bunch of mini columns for each, cells aligned above that or below it, and that way you'd activate one mini cell per column and achieve a large space. This is nice because it fits what we're doing in layer 4, and but I didn't have a mechanism to make it work. But it did give us a large space, which is what we needed. So then, as I said earlier, oh damn, I don't know why that happens to me all the time. So I just scroll around and it. So let me just tell you, I was working on presenting how he's going to get this stuff to work, but instead, I want to show you a new paper that that came out. It wasn't even that recently, but it was. I wasn't aware of it. So this new paper was from the Moser lab, or Sensola and Moser, and Moser was the guy who first discovered grid cells. And this is a 2016 paper, Grid Cells and Spatial Maps of Entorhinal Cortex and Hippocampus. And here in this paper, they say something completely different. They say, yes, grid cell modules are these one by one. They give the dimension as a millimeter by millimeter, which is larger than what Tank showed. So I don't know what to make of that. I mean, Tank showed there may be like a half millimeter by a half millimeter or something like that. They're selling millimeter by millimeter. And they have all these properties that we talked about. They get larger in scale as you go up, whatever. Oh, I'm sorry, this is it. But what these guys said is, no, these things are not independent. They said they overlap. And I'll show you the text in the paper. So if I pick a point in the grid cell in the entorhinal cortex, like this point right here, there are several sets of grid cell modules that are underneath that point. So if I sample down through the neural tissue here, I'd be reading from multiple grid cell modules. Now, not 20, but not one, maybe 3 or 4, something like that. I've shown 3 here. And here's the text in that paper. So it's, so here's in this first yellow highlight says individual modules occupied extensive portions of the medial entorhinal cortex. We found on average a module span greater than 1 millimeter of the dorsal ventral axis. So they're actually greater than a millimeter in extension. I've showed it as a millimeter. Then in the next green one says there were extensive module overlap in the intermediate intermediate to ventral parts of the MEC, such that any MEC location cells from several that at any MEC location, cells from several modules could be present. Grid cell modules were found that cut across cell layers. Cells that were part of one module were found in several layers. I don't know what to make of that. But that's the critical point here. It says that any location, cells from several modules would be present. Not one, several. Then the next yellow one says there was no discernible topography along the medial lateral axis. That's the other axis. That's the one that's going across a different direction. And instead, modules extended across the medial lateral axis about a millimeter, which is the limit of the recording. So it could be more than that. And
I got a
then
theory.
one second, let me just finish it up. Then they could estimate the number of distinct modules in the animals to be in the upper single digit range. So that was consistent with what we've heard before. Yeah, Scott, go ahead.
So the modules have different spatial scales, right?
Yes.
So if you wanted to have a way to dedicate more resources to one scale over another, you would want to have cells that can kind of stride both of them and say, okay, now I want to be more attending to this level of spatial scale. Hey, you guys on the boundaries that you're kind of also able to do higher spatial scale or lower, let's pull you in and basically recruitment, like an attentional recruitment.
Right. It could be. Although that would be getting around the benefit I saw of this is that I am now able to form a representation that spans 3 or 4 grid cell modules. And if I only focus on one, I'm going back to my impoverished representation.
Mm-hmm.
Right. Like, but I see your point. I mean, yeah, it could be doing that. But I was excited for a different reason. I was excited because I wanted something that I could use grid cell modules for, but get something a higher resolution than I could have a single module.
And just to clarify the language about the grid cell modules kind of going across layers, do you know when they recorded the activity? So could they simultaneously see like a grid cell at scale X, a grid cell at scale Y, and a grid cell at scale Z sort of like stacked on top of each other?
That's the implication. Yes, that's
Yeah.
what they were saying. Whether they are stratified, I didn't get. Literally, I ran across this paper yesterday as I was stopping the work, and so I haven't read it carefully. But I mean, the language I just read you, this language, says there's extensive overlap. So that would mean if I put my probe down through the surface, I would run across cells of different scales. They were found to exist across cell layers. Cells that were part of one module found in several layers. So I assume that meant they're all kind of intermixed because it's not like, oh, the top layer is going to be, it's not like a bunch of, stratified layers where the top is going to be one scale, the next layer down, the next scale, next layer down, another scale. This language suggests they're kind of mixed together, but I'd have to read the rest of the paper carefully to see if I could deduce that. But my assumption at this point was this: all I could say is like, hey, if I'm at some point along the surface and I dig down, I'm going to find grid cells that that are actually part of not just different scales, but different grid cell modules. They will anchor separately. Therefore, I have a higher representational space. But whether they're stratified or not, it sounds no here, but I don't know.
It's also kind of
Is that
interesting how this partial overlap, like it's not the, as you're kind of saying, I think it's not the like extreme version of like 20 modules in one on top of each other. But
right?
it is some amount of yeah overlap. But yeah, because I feel like you've also talked recently about like to what degree columns might kind of overlap and almost form like larger.
Right,
Um,
right, right. Exactly. So I was working on this idea. I don't have a good picture of it. That each column had a single grid cell module, but maybe those grid cell modules sort of lockstep together. So a bump moving from one would slide into the bump moving for the other. And and so when you anchored one, you anchored all. Of course, they'd only be one grid cell module, so you have a very limited representation. But at least it was a way of thinking about, hey, I can lock them together so that at the level of a single grid cell module, they'd all be agreeing that we're all locked, we're all remapped at the same point. Um, so that was the idea I was working on, and this bumps it up a bit. This says, hey, that idea is maybe somewhat still valid, but I don't just have one grid cell module at any point in time. I might have three or four of them that are overlapping. So let me just go sort of walk through some of my thinking about that. Um, okay, so what are the implications of Cortex? But for starters, I'm gonna assume we have like color columns or millimeter square, like I've argued recently. There's various thinking about this, but there's a number of people say, yeah, they're millimeter square. And then I'm assuming there's a millimeter square of grid cell modules, just like the grid cell modules are also millimeter square, just like we see in the entorhinal cortex. Uh, then I had to make an assumption. How many unique locations can a single grid cell module represent? There's, you know, how many different cells does it take to map out, that have some unique locations? I took a total swag at this, 30. Okay. Like, like there might be 30 different bump locations. It's complicated to think about, but I just picked the number. I think this is on the low side. It might be higher than this, but picking a low side, I thought. Then I pick also another. I said, what if, as we see there, that they overlap as in the entorhinal cortex? So imagine under a particular column, I had three overlapping grid cell modules. I mean, that doesn't quite fit with my millimeter squared up there, but let's assume that was the case. And we assume that those grid cell modules map independently. So every cortical column, instead of just relying on one grid cell module, might have three different ones. They don't have to have different scales. We see the different scales in the entorhinal cortex, but that may not be necessary here. I just need. I'm just going from moving from one grid cell modules to three. Okay, and then I said to myself, okay, so what is my representational capacity? I don't have 20. I got three. So what can I do there? You can say, well, um, what is the number of locations I can represent with three grid cell modules like that? So it's basically 30 to the third power. I mean, instead of one module, I have 30, two modules, I have 900, and three modules, I have 27,000. So this meant going from like 30 unique locations to 27,000 unique locations, which is not bad. And so this is the number of points for which path integration would work. Meaning I could path integrate these three separate grid cell modules, and any point in this space would be path integrated correctly, which is far better than I had before. It's probably not enough, but it's far better than I had before. Um, which is like if I only have one grid cell module, it's really limited. But three, okay, that's not bad. It's getting someplace. So this is like you point out, Neil, this is like an in-between result. It's not ideally what I want. Um, and the SDRs for these locations, one of the issues here is that these SDRs for these locations would not all be unique. That think about it this way: I have three sets of 30, um, and um, and so at every 30th location, I'd have two of those grid cell modules would if I'm thinking about it correctly, you could have a lot of it wouldn't always be unique. We don't have the assumption of total
but I guess
uniqueness.
it kind of makes use of the SDR. Well, yeah. So I guess if we use the sort of what HTM does in L4, that resolves a lot of the uniqueness stuff. But the problem with that, I guess, is it's kind of directionally sensitive, right? As in, like, if I said, if you imagine the grid cell that's active is kind of like the mini column that's active. In some sense, that we can still get a unique representation if we predict a set of subcells in it, and
Right,
then they predict a set of subcells. But that
right.
prediction of the other set of subcells, that needs to be learned. So we kind
So,
of need to go through, at least
so
in traditional sequence memory. And so we need to kind of like visit the same path for.
well, so let me just put my interpretation on same word, same idea you're talking about, but different words. So, uh, before I had no mechanism for doing path integration of the individual cells. When I assumed that there was mini columns above a single grid cell module, I didn't really have a path integration method to update the correct cell. I mean, we had to, but I didn't, and I talked about, you might remember, I talked about how these directionally sensitive cells were, to create grid cells, you needed these cells at different phases and maybe, but there was no way of re-anchoring it was, we could go into details. It was really complicated and it didn't have any work. So the current thinking, I'm doing something like the following. It may be a hybrid approach. Maybe the mini columns in layer 6 are really representing the coincidence of these three grid cell modules, so, or something like that. So I have one representation, which is this three grid cell module representation, and it does path integration. And it's not as high resolution as I'd like. It's not as many as points as I'd like. Um, but it does work for path integration. On top of that, I could still have our mechanism for mini-coms, and I could make a very unique SDR out of each one of these 27,000 locations. That I could say, oh, I can create a very unique representation if I use a mini-com, but the path integration wouldn't work on those cells. It only works on the mini-coms. This is similar to, maybe if I can just jump down here for a second. Um, uh, Here. Here's a picture I was drawing. This is how I was thinking of it recently. Whereas we had layer 4, we assume that layer 4, the mini columns are driven by the sensory feature detected by the retina, for example, right? Feature from a sensor. It's very low precision. It's not unique. Um, that's the only thing when we drive this layer 4, that's what we get. And then we pick unique cells in the individual mini columns that are active, and that's forming unique in context. But if I get a new input, I can't predict these. I'd have to have some other context to tell me what they are. And the same thing was thinking down here. I could have there could be mini coms in layer 6A. Each one corresponds to a single grid cell. And then I would pick a unique cell, and this would be a unique version of that location. Um, and all I'm pointing out is just I could predict, I could do path integration to say where's the next grid cell that'd be active, but I had no ability to do path integration um, to say what's the next set of cells that were active, right? And now the idea of this new paper introduces the concept that this layer may be must be much better than we thought. This layer might say, oh, it's not just one of 30 locations. Maybe somehow it could represent a much, a richer space, but I would still have this problem if I wanted to represent more uniquely with cells. I couldn't do path integration here. So I could do, it's like I get halfway there. I say path integration will tell me a lot about what I should be expecting to see, but it may not always be unique. And but it's a hell of a lot better than I had before. So it's sort of a hybrid in between. Like we can do much better path integration easily using multiple grid cell modules, but I might still have a higher and more sparse representation that I can't do path integration over. So I'm going to have to leave it like that, unless you can think of something better that I can think of. Yeah, this all happened just in the last, few hours well, end of the day yesterday.
Yeah, no, that's interesting. I mean, in terms of it being kind of, yeah, hybrid approach, it feels like also it's getting a trade-off in that, yeah, we have some path integration, as you say, in the higher one. We could still learn like the active cell in the mini L6A mini column could still learn that, okay, after you move, and so the bump moves along the top, I'm going to predict X cell in this other mini column. Um, but that
But I
that
don't know
that's a
how it,
that's a
how would it,
specific learned
how would it,
path. So
how,
that is not true path integration.
right, right. I could learn a
But that's why I'm
sequence like,
saying
oh yeah,
it's more like a hybrid. So that would be almost more closer to like the displacement stuff
right,
we did
right,
before, where like
right.
maybe some of the lifting is done with path integration at the top, and maybe some of it is these more learned um,
Well,
associations.
the real learns associations are shown here in this arrow between layer 4 and layer 6A. This, in some sense, um,
Oh, so okay, I
uh,
see. Yeah, so you weren't right. You weren't thinking of like
right.
recurrence within the
This pattern
L6A.
here, you know, it's sort of saying like up here I have a pattern in layer 4. This is the mini columns that are active. That isn't unique enough. So, in some sense, all the cells are sort of bursting in there. But when I project it down here and I try to associate it with another pattern down here, these things will resolve each other. It's like um, knowing a poor representation up here narrows down my poor representation down here. And I haven't worked through the numbers yet, but it might be sufficient. It might be sufficient to say, like, hey, given I have a, one of 27,000 locations here type of thing. Again, it is all hand wavy. Um, and it's not as accurate as I want, but I have a feature up here. It's not as accurate as I want because it's a feature out of context. But if I take the feature out of context and combine it with the location, which is not great but pretty good, the intersection of these two may be unique. That is, I may be able to achieve all the things I need to achieve. It's just I have to think about it mathematically here. That is, you know, uh, given I have a reasonable but not good enough representation here, that my raw feature may be able to say, yeah, I know exactly where you are. You have to be here because we're combining those two. And similarly, if I have a feature, if I know exactly where I am here, then uh, I could predict exactly where I am up here because we've learned that association. Uh, anyway, it's a it's just a new avenue to think about this, um, and I haven't thought about it enough. Let's go back to the picture. I think is important here. It's this picture here. Let me go here. Uh, what are the implications of thinking about a system like this? And I've just started to scratch the surface on it. And it's weird that why did other people not show this? And why did other people, when I talked to Tank, I asked him specifically, is there any overlap between these? And he said no. So how do you explain that? I don't know. I don't know what to say about it. I kind of believe the Moser paper because it's um someone like Tank might have missed it. That's reasonable. Or he might have had some bias not to look for it. That's understandable too. But if someone says, I saw it and this is what it looked like, that's more believable. So the experimental paradigm might have missed it and showed you this, but a different experimental paradigm shows you this. It's kind of hard to dismiss it. And it's coming from Moser, who's very precise and accurate. Um, So I just thought that was fascinating. And of course, this also sort of means that somehow one column might bleed into another column. And so, there's this idea that there's sort of maybe, there may be some sort of lateral coalescence that's going across the cortical sheet. Because one column's partial knowledge spills into another column's partial knowledge, which may then everybody you may have some sort of rapid, if two columns that are separated by some distance and are not, you know observing different parts of the object, then maybe there's could be like some sort of coalescing in the grid cell module layers. They all sort of come to some consensus. I don't know, but it certainly
Yeah, which
opens
to
up
be
a whole
fair,
whole different way to think about things.
I feel like that fits also with a lot of like neurophysiology evidence, which of course you have to be careful how you interpret it, but like outside of barrel cortex, most cortex doesn't show at a like response level uh, super obvious kind of delineation. Like I know
There's
uh,
no physical delineation. Is that what you mean? There's
yeah,
no,
there's definitely no physical delineation, but even in Mountcastle's like showing on the paw and stuff like that.
yeah.
Yes, you kind of got these discrete jumps, but also there was often a lot of overlap.
Well, there was
Uh,
overlap, right? He showed that too, right? Right. So there's overlap,
whereas
but
it's
he's
only in barrel cortex where it's like, yeah, whisker, 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 like totally separate.
right, right, right. One of the reasons I like Mountcastle's idea. I should stop sharing if you want. One of the reasons I liked his ideas is because it's kind of hard to imagine how it could be otherwise. It's kind of hard to imagine how a continuous sheet would learn discrete objects. You know what I'm saying? You kind of have to parcel it out somehow. It seems like I don't know what it means to have a continuous object that model of something. It doesn't make any sense to me. But the idea that you could think about a cortical column is so one column is separate than another column, separate than another column makes a lot of sense. And there's various ways you could do that. So I guess I'm still sticking with his proposal that a square millimeter cortex is processing input from one patch of sensory, and then it's not like a fuzzy boundary. It says okay, one patch of sensory cortex. The next column could be having an overlapping patch of sensory region. Sensor.
Right, but it's
That's
still a
not that.
different sort of
It's
model
still
we're
a different
referring
one,
to.
right? Right. But now we're blurring the now we're saying, oh, but yeah, but but now they're sharing. Well, I see what you were saying a moment ago. If you just take Mountcastle on the surface, even the simply sense it would already have two. I don't know what the hell. I have to think about this. I don't know. Anyway, the idea that they're overlapping in sensory space might be meaning they're overlapping in. I think is what you're saying. The fact that
Yeah,
they're
yeah,
overlapping in sensory space, they may be overlapping in grid cell space.
right.
I didn't catch what I got it. Now I get what you're saying. I didn't get it. Now I got it.
And even like
Yeah.
I think when they try and again you have to be careful, I guess, how you interpret it, but like even when they look at like how object categories are represented or whatever, and like like there just seems to be kind of like more like a smooth, continuous change as you go across. Um, yeah.
Is that
So
right? Um, is that
but
true?
yeah, but
Uh,
of course that's if you kind of understand neural responses as more kind of like grandmother cells responding to certain objects and
I
stuff. So
see.
it's
Right,
not
right,
necessarily applicable, but
right. I still think, I'm not giving up on the column hypothesis at all. I think that's not even thinking about that. Um, but this overlapping part is sort of that's kind of new in my thinking. I assume these things are discrete, they're separate. We've talked about them as individual entities, and that they didn't overlap, even though at least from a processing point of view. And yet, these questions, these problems that we're trying to deal with, assume that they're not discrete. They are interacting in various ways, and this is just one more way they could interact. Um, and I think it's sort of eye opening. It was like, oh, this doesn't throw away anything we talked about columns. It doesn't throw away anything we've done. It just says, oh, how might they interact, and how might we represent locations in a column. And again, Monty uses a different mechanism completely, so this doesn't impact Monty today. Um, I'm going to leave it there. I have a lot of other stuff I've presented, but I'm not going to present it because I was like going down another rat hole. So
nice. Yeah, I think that was really interesting. Yeah. So I thought it might be helpful to kind of um, paraphrase, I guess, what you were saying at the end. Would this be like a fair representation that instead we kind of have these sort of like three, and maybe you could even show them kind of like because they're
right,
coming from
right,
different
right,
parts,
right, right.
but they're kind of meeting here, and so between them they give us some like a more unique location coding, and maybe that's kind of enough with this whole recurrent system
Uh,
to narrow it down.
yeah, it's a general idea, but I'm not sure I'm not willing to say those details now. I think the
Yeah.
key thing here is you don't want your red and green and blue dots to be independent. You want to be sampling from the three of them. Um,
Well,
uh,
uh, as in they should be able to re-anchor independently in order to
They
create
they
unique code.
right, right. But
But
to get the
as in
benefit,
like the L4 cell should be getting input from all of them, kind of.
the benefit of having the three grid cell modules is that the cells are going to respond to the combination of the three. Is like a cell would say, all right, I'm responding when all three of these things are, these three, this green, this red, and this blue are active.
Yeah, you mean a cell up
Uh,
here in
no, and
L4.
even the cell down below, it's like, well, someplace, I don't know,
Yeah,
Niels, I don't know. I have to think about it.
yeah.
This is all fresh. It's like, in fact, now I'm looking at it here, I would try to write, I might show, I would try to think about what are the implications if I had three columns and and they overlapped in sensory space and they overlapped in grid cell space. Because that's what Mountcastle was showing, right? He was showing that they overlapped in sensory space, meaning they had overlapping receptive fields on the sensor. I have to think about what does that mean. I mean, maybe I would draw three columns that are independent but are overlapping in sensory space and overlapping in grid cell space. I don't know how to do that yet. I have to think
Yeah.
about what the right picture for that is. But I think someone I guess the answer is I don't know what it means. That was a good first stab, Niels. But I think we're gonna need a bunch of thinking to think like, oh, what are the other ways to interpret this? So all I'm saying is that we started out with one a long time ago. We said, okay, grid cells need a lot of grid cell modules. That'll be perfect. There aren't a lot of grid cell modules. They were separate. Can't do that. There's no overlap. Blah blah blah. So we went to other extreme. One grid cell module with mini columns, and that created a very large space. But we didn't have path integration working. There was no way of influencing one to the other. Difficult re-anchoring or remapping. So a lot of problems there. And now there's something in between. It's like, oh, we have a bigger grid cell space, but not as big as maybe we want, but it's bigger and big enough that it might be good enough for the inference that we need to do. And it also might lead to things like, Rami, you'll be happy to hear this, it might lead to things like, hey, maybe there's ways that these representations do overlap. They're not true SDRs anymore. They're not like really big SDRs because there's only like three grid cell modules maybe. Um, but it's sort of an intermediate representation where it's sparser than a single grid cell module, but it's not as sparse as a true SDR. But there's be more overlap in these intermediate representations. So maybe there's some way of interpreting them as partially semantic and partially SDR. I don't know. Who knows? It's crazy. Um, So that's it. Basically, a new paper which throws into question a lot of stuff we're thinking about.
Yeah, nice. Yeah, and also someone interrupted, I don't want to hog, but I just wanted to ask, could this also expand the representational space, I guess, if you use the different kind of submodules or wavelets, like if the sort of scale, not the scale, the sort of phase between them, if that was different, if that could re-anchor differently, it
Well,
feels like that could be another dimension,
right, but
maybe.
I don't think that's the case. Um,
Okay.
the way that Tank described this, and it makes sense theoretically to me,
Yeah.
is these different, I think, did he call them wavelets or phaselets? I think he called
Or
them
phaselets, sorry.
maybe phaselets or something like that. I don't remember what his term he used. Um, they're just sort of duplicates of each other, which makes sense. That when people describe um, uh, when they describe uh, a grid cell module, they're really talking about just one of these little phaselets. Uh,
Yeah.
but he says there's like six of them, and that you need that because it's like a bump of activity. And first of all, you need more than just like one set of cells representing something. You need some redundancy into the whole system and some more cells that could mean something. Um, but he's describing them as, like the yellow cell here, all the yellow cells they're exactly the same. It's just like, so instead of one bump moving around on this, you got six bumps moving around, and as one bump moves off to the right, off the right edge, another one appears on the left edge. Um, so
Yeah, like perfect.
there was no
So
additional
yeah, they
representation.
always maintain a constant phase
Right,
relative to
right.
each other.
There's no additional representation capacity here, and having six of these, it's more like robustness and redundancy, and it makes the synapses work better. Um,
Yeah.
and these little black lines he shows there, separating the
sorry.
phaselets.
Yeah.
Those are just arbitrary. I mean, he just drew those in. There's nothing
Right.
physical about
Yeah.
that,
There's
right?
no little thing in the brain.
Right.
You're
He's
right.
just saying, he's just pointing out like, hey, this basic thing repeats, and here's the line I put to make it repeat. He could have put the lines elsewhere and achieved the same result. So just they're not really discrete things. It's just
Yeah.
like there's
Okay.
a bunch of cells that are all sort of bunch of bumps that are moving around the same surface. Um, and I asked myself how many different, I said 30. Remember the number 30? I was asking myself, well, how many different locations could you represent in one of these phaselets? And it's complicated because I was looking at these are individual cells he recorded from. These aren't all the cells. These are just cells he recorded from. So, they poke around, they find a cell, they characterize it. So there's clearly more cells, many more cells than he's showing here. Um, and uh, and so I'm trying to get, and so I said, well, how many could you fit in there in one of these phaselets? And then of course it's a bump of activity. So it's not like one cell's active and then the next cells become active. It's like a few cells become active and then the next overlaps with the next few cells. So I just took a stab and I said, well, it looks like I could easily fit, 50 cells in there or 100 cells in there. And maybe the unique bumps are going to be less than that. So it was a total slag of 30 to say how many different locations could you
Yeah.
represent there. But, I had to sort of back of the envelope calculations to see if it made sense at all. Yes, Scott.
I thought one of the authors there was Densola.
Yeah.
I know him. I could definitely get him to do if you want to bring him in sometime to ask him questions.
That would be great.
I could probably arrange that.
Oh, I'd love to have that. Right. That'd be lovely if you feel comfortable doing that.
Yeah, he's a super cool guy.
That would be neat. I love that. Is he in Moser's lab or is he in a separate lab?
He and his wife both did their postdocs in the Moser's labs.
Are they
So
still there?
I said they started their own lab, but they're still in
Okay.
Norway.
So they're separate labs now, right? I would love to have a chat with him about this stuff.
Yeah.
It was
Okay.
another interesting question thing about is in the entorhinal cortex is all these scale differences, right? If you go one grid cell module to the next and to the next and next, you get this. Everyone is going up in scale by this factor of square root of 2, basically. And so I jumped to the conclusion like, hey, that's similar to the way we see a scale change in V1 and V2 and V4. And I said, well, maybe what's going on in the entorhinal cortex is paralleling that construction. And so I started thinking like, oh, in the medial entorhinal cortex, you might be learning um, compositional structure, which I think you probably would have to do. Anyway, but then in the cortex, I wouldn't assume that if I'm in V1 and I take adjacent columns in V1, I don't assume that the scale of the grid cells are going up by a factor of 2 every time I go over a column. But so we wouldn't assume that's the case in like V1. However, it's interesting. Even in V1, if you
I was just
have a
gonna say it's
lot of
kind of
scale
interesting when you think
differences.
about eccentricity. It might not be a
Right,
crazy.
right. So as you keep going, and once you get out of the fovea area, the scale does change in V1, right? It gets really larger and larger and larger. So we shouldn't have our thinking constrained by that idea. Just because we see that scale increasing in the entorhinal cortex doesn't mean that that concept has to always be like that. It's maybe the important thing is you have overlapping grid cell modules, and sometimes they change in scale, and sometimes they don't.
Wouldn't you need the scale change to get the unique locations when they overlap?
No, you don't. It helps, but you don't need that. When a grid cell module remaps, there's two ways it remaps. One is which cell is active at any point in location. So that let's say 30 different points, but they also have slightly different orientations. So they change their orientation a bit every time that they remap. So that brings another multiplying factor to the whole thing. So yes, it's helpful to have different scales, but you can get pretty big representations. If I'm saying I'm just assuming there's 3 or 4 overlapping grid cell modules, um, the scale change isn't really going to make much of a difference because each grid cell module will have a different cell active and a different orientation, and the combination of those is sufficient to really represent everything, I think. Maybe if I wanted to do like, 20 overlapping grid cell modules, I might need scale changes. But for 3 or 4, I don't need that. I just need different cells and different orientations, and that's a lot of combinations. So there's a lot of different ways you could, in some sense, that tells you how many reference frame spaces you have. If I had 3 grid cell modules, um, how many different reference frames could it represent? It depends on how many different ways you can re-anchor or remap those 3 grid cell modules. So uh, you might think of it as something like each one has like 100 different remappings. Let's say 30 times 3 different orientations. So 30-point positions on 3 orientations. For 100. So that'd be 100 for each one. So then you'd be like, a million, 100 cubed, a million, right? Yeah, a million different maps, but you only have 27,000 locations. So it's like you can have a lot of different remappings, but you're still limited to the 27,000 unique locations. It's just,
And I
I don't know, it's
guess,
just different ways that they
yeah, I
think
guess
about
you
it.
lose a bit of that because the overlap's only partial, so you don't have 3 on top of each other, but or maybe you just lose some of the redundancy. Um,
I don't know. I mean, it's weird because if I'm in a single grid cell module, I'm moving across that grid cell module, a single cortical column, excuse me. If I'm in a single cortical column and I'm moving across that cortical column, as I move across, I'm intersecting different grid cell modules. It's from different
yeah.
places. I was like, weird. I don't know. Again, it's like, I remember when, I'll get to you in a minute, Hojae. I remember when I first learned about grid cell modules, I was so damn confused by them. I mean, for a month, I just couldn't even get the concept. It was so confusing to me. And so now I'm changing the whole thing a bit. I'm like, it's going to be confusing for a while, I think. Again, that would be my guess. Yeah, Hojae.
Uh, yeah, I just wanted to kind of share the paper that the Sensola and Moser. So I think the paper that you shared is review paper, and I tried to go into the primary paper that it cites to kind of make that claim. Let me see if I'm sharing the right thing. So, I mean, I think they're citing their own paper from this. The Edwin grid map is discretized, which I think we went over this when I was doing like a grid cell review like a long time ago, back in 2012. But if you go down to look at, I mean, so first of all, the kind of
I'm sorry. Are you saying this is the
analogous.
one that shows that they're overlapping?
Yes, yeah. I mean,
Okay.
you can see from here that like, well, they recorded, well, they found kind of 4 modules in whatever area that they recorded, and you can see that these 4s are overlapping.
So we read this paper.
Yes,
I
we
didn't.
did.
I didn't pick this up. How did I miss this?
Wait, well,
I
I
think
don't
it
know
was.
if
Uh,
Hojae did, right? You
Okay.
did
yeah,
when you were
I
reviewing
don't.
yeah.
the.
If I read this
Uh,
paper, that would have jumped right out at me. I would have done, wow.
Though I
Okay.
think, I mean, their wording is kind of weird. So first, like I think we focused on this, and this was a little bit misleading in the sense that so like each of the black dots are grid cells, and they found they can group the grid cells by like the spacings, and we thought that these were kind of separated spatially as well. But then what they found is that if you actually these are just like indicating what the spacings are, which like scale by roughly square root of 2 according to them. Um, but when you actually plot where they are in the dorsiventral like locations, whoops, then you can see that these box plots are like significantly overlapping, which is I think represented in um, F here. But I think in
F,
text they
F,
say
F, F seems to imply, if I don't know how to interpret that picture, but it seems to imply that it's showing the different modules at different depth of the Toronto cortex, and that they're mixed together. Is that the picture that says, hey, these things are all mixed in different layers? Is that what that's showing?
that's by interpretation. Let's see. So F left, not that. Do. So they don't have an explanation for this. Oh, right. Outline of each module extrapolated from flat map, same scale. Okay, well, that's not really helpful. But in the text, they say in animals with more extensive dorsiventral sampling, I'm reading from here, and more than one grid module, values for grid scale and grid orientation are similar at widely separated medial lateral locations. I think again this is supporting the mixing. But then I think the unfortunate set this, which indicating that within the recording area of the medial MEC, the modules may be organized as horizontal bands parallel to the dorsal border of the MEC, which I think maybe this may have misled us to think that like the modules are separated in like little bands instead of being kind of
Well,
mixed together
I
like
don't
that.
know. I mean, I don't think I read this paper. If I did, it's
Okay.
scary. But can I see the title again?
Yeah, a nature.
That title seems familiar.
Right.
But just like yesterday when I read that other paper, the summary paper, it jumped right out at me like, oh my God, they're overlapping. So I think if I read this carefully, I would have caught that. So so I don't know if I read this carefully, but that looks like a great paper. I want to can you just post
Yeah.
that so I can read it
Yeah,
carefully?
yeah.
I'll read.
Yeah.
That seems really important. Thanks for bringing that up.